Good morning, guys. Can you hear me well?
Yes.
Good. I guess my internet is working. All right. So what is it, Wednesday? We're working on chapter 4. We're working on three-dimensional structure of proteins. So last time we finished by discussing the Ramachandran plot. So let me switch to, actually, before we do that, I just want to remind you, I sent an announcement that I released a practice test. It's a short one, just for you to get familiarized what kind of problems you may expect to see on the actual test, on the first test on September 30th. So you can see that the practice problems kind of composed of material from lecture, material from sapling. So have you been able to look at it yet? Anybody has looked? No, nobody has looked yet? I just want to make sure that you can actually see it. Well, like I said, this is my first semester with Canvas, but it looks like you should be able to see it. So can you do me a favor after this lecture? Can you look at it and see if you don't want to do the problems yet, that's fine. But at least if you cannot see it, if it's not showing on your supplement course, on your biochemistry course page, please let me know right away. Okay.
We can see it, Dr. Korniko.
Yeah, it's there.
Yeah.
Okay. All right. So again, as usual, if you have any questions about it, put them and throw them in the discussion section of Canvas, so it's useful for everybody. Much more useful than just a separate email to me. It's more useful for everybody if we all participate. All right. Share screen. Share screen. So Ramachandran plot. Going back to the zigzag orientation of the peptide chain, right? So we talked about the two dihedral angles, phi and psi, right? So we talked about this omega. Omega is 180 always, right? In 99 point something 6% cases, it's going to be 180. And we will look at the specific case when it's not like that. But in most cases, it will be 180. And so phi and psi, remember it's 180 if it's fully extended, right? So in other words, if it's trans, so phi is the angle between the nitrogen and the alpha carbon, and psi is the angle is between the carbonyl and the alpha carbon. All right. And here is, you can see we talked about this, right? So in most proteins, there's very limited distribution of this or combination of this phi and psi, right? Mostly localized to this upper left corner. A little bit here, which represents alpha, left-handed alpha helix. This is right-handed alpha helix. And so here is the distribution of all amino acids in the enzyme pyruvate kinase. This is pyruvate. Oops. Let me start writing this down. Pyruvate. How do you spell pyruvate? Y. Pyruvate kinase. You can see that all the amino acids except for just a couple outliers. All the amino acids occupy these areas which are acceptable combinations of phi and psi, right? All the other ones will lead to some intolerable steric hindrance, which will prevent the protein or this enzyme to adopt a particular combination of phi and psi.
Let's have a quick question then.
Yeah.
So to interpret a Ramachandran plot is simply comparing the pi and psi angles to what is the most optimal combination of two, and that's what's located in the blue areas.
Yeah, it's not even like the most optimal. Most optimal will be like the dark blue, right? The light blue is it's not the most optimal, but it's still acceptable, right? Whatever you see here, like in this area where it's blue, where it's white, there's nothing here. It's just unacceptable. So in other words, the combination of phi and psi, so this leads to such strong steric hindrance that the protein will never adopt this kind of conformation with this kind of combination of phi and psi, right? So I would say the most optimal is dark blue. Light blue is still acceptable, and no blue is unacceptable.
Okay, thank you.
Mm-hmm. All right. So secondary structure. So secondary structure will be the local spatial arrangement. So usually these are short segments of protein backbone, right? So there'll be the secondary structure. So the two most common that we will talk about, alpha helix. So this will be stabilized by hydrogen bonds between nearby residues, and beta sheet, stabilized by hydrogen bonds between adjacent segments, which may not be nearby. So we'll see that as well. And if you have irregular arrangement polypeptide chain, if it's neither of those two, and it doesn't particularly adopt any particular kind of order, we'll call it random coil. Random coil. So you can see here, this is the combination for different secondary structures, the combination of phi and psi. So these are two numbers, good to remember, minus 57, minus 47, easy to remember for alpha helix. Let's go back to our Ramachandran plot right here. Where was our alpha helix? Here's right-handed alpha helix. So you can see here, this area, right? So minus 47 for which was phi, which was psi? Phi was minus 57, psi was minus 47. So minus 57 is somewhere here, right? Minus 57 for phi, and minus 47 was somewhere here for psi, right? So that will be the right-handed alpha helix. And the other ones are also listed in this table. Just for reference, obviously I don't want you to memorize these numbers. All right, so let's start with alpha helix. So here the helical backbone is held together by hydrogen bonds between the backbone amides of the N and N+4 amino acids. So let's look at that. So if this is the NH, the amide, NH of the peptide bond, right? Right here, the white little sphere is hydrogen. And then we go around this axis, right? So if this is amino acid N, then this carbonyl will be N+1 going to the side chain, N+2 going to the side chain, N+3 the side chain, and finally you hit the carbonyl of the N+4. So this is the fourth amino acid, and you can see here there is a nice hydrogen bond here, right? And you keep going, and then you hit another 4 amino acids, another hydrogen bond, and so on and so forth. But each NH, so this NH, it's not actually. It can be seen that actually, because of this axis, some of the hydrogen bonding are hiding behind this this rod, right? So it's not really visible. But each amino acid, each NH of a peptide bond will be hydrogen bonded to the carbonyl group of amino acid, which is 4 amino acids ahead of it, right? And so in general, the distance will be the of one helical turn is 3.6 residues or 5.4 angstroms, right? So this is carboxyl terminus, this is amino terminus, and so the peptide bonds will be roughly parallel with the helical axis, and the side chains point out and roughly perpendicular. So you can see here, these are side chains. This one, then this one, they're kind of sticking out, right? And in fact, I even have a picture. Let's see, yeah, right there. If you look from the top of the alpha helix, you can see here, this is the alpha helix itself, where the amino acids kind of turn around, but the side chains are sticking out to the sides, right? So the side chains are never in a way, which allows for multiple amino acids to participate in the formation of alpha helix, right? So if the side chain, they play a role, and we will look at it in a moment, but they don't really interfere with the formation of the alpha helix because they are on the outside, right? So if you think about it, it kind of looks like it doesn't really matter what they are. It does matter because they're still close to alpha helix, but they don't break up the alpha helix right away, right? So they are kind of a certain distance away from the alpha helix. All right, so where are we? So what's left-handed helix, right-handed helix? So there's a stem, right? So that's what they describe in the textbook. You can use this. The way I look at it, if this is the direction of propagation, right? And the alpha helix turns in the clockwise manner. Then it's right-handed. And this is counterclockwise. If it's counterclockwise, it's left-handed.
How will we be able to tell that? How will we be able to tell that they're turning in when we see a molecule?
How you'll be able to tell? Well, they are turning. They're twisting around in the helical fashion. Yes.
Well, like I mean, like on like the prior slide when you show the alpha helix, it said it was a right-handed helix. How will we be able to look at that and say, okay, it's right-handed without it being labeled?
I still don't understand the question. So if it twists in the counterclock fashion, you know it's a right-handed helix. So same thing here, right? If you position yourself at the bottom, this is your eye, like here. Remember when we did the RNS configuration? You position yourself down there at the bottom, and you look in the direction of the propagation from the carboxyl to the amino terminus, right? Then you can see there that it's turning in the clockwise direction. So it's going to be right-handed. And that's the simplified description of that.
Okay, thank you.
Yeah. All right, you can see that actually the inside of the alpha helix is quite packed, right? So not so much can get inside, and the side chains are on the outside. So something for us to think about. So if this is the amino acid 1, remember it's going to be hydrogen bonded with the amino acid 4. And let's say if there's a side chain here, what else? Okay, 1, 2, 3, 4. It's going to be hydrogen bonded, right? So 5, for example, is going to be hydrogen bonded with 8, right? 9 is going to be hydrogen bond. 6 is going to be hydrogen bonded with 9. So basically n plus 4. Now there is something in this picture that I haven't mentioned. What else do you see? So you see these spheres for representing amino acids going in a helical fashion, right-handed, alpha helix. What else is here that I have not mentioned? What else do you see on this picture?
Peptide bonds?
Well, they're not really shown. No, not peptide bonds. There's something here that I have not mentioned that is very important that we're going to discuss, spend another 5 minutes talking about. What about this? What do you think this plus and minus mean?
The charges of the molecules.
Not molecules. Peptide is one large molecule, right?
The charges of the amino acids.
The charges of the side chains on amino acids. That's right. So then tell me, if we talk about stabilization of alpha helix secondary structure, so not all alpha helices will be stable, right? So we're talking here about whether the protein segment will adopt alpha helix or will the alpha helix be unstable. So what amino acid will you position in the N and down the line, N plus 4? Which amino acid will you put here? Which 2 amino acids will you put in these positions to stabilize alpha helix?
A basic and an acidic one.
Yeah, so what's your amino acid 1?
We can do lysine,
What's your amino acid 4?
aspartate.
Aspartate, perfect. So if you see something like this, and you may see this on the test, right? If you see lysine, if you see X, Y, and aspartate, and it keeps going, right? It keeps going this side, keeps going on that side. It's highly likely that this particular segment, well, you need to have more amino acids for alpha helix, but it's highly likely that this will adopt the alpha helix. Favorable. Now, what about something like this? Glutamate, X, Y, aspartate. Will this contribute to a stable alpha helix?
No.
Yes.
So somebody says yes, somebody says no.
It'd be turned the other way.
Yes, so basically now here we have negative charge and negative charge. So these will repel each other. They will not like to be next to each other. They will not want to be hydrogen bonded because the side chains are both negatively charged. So this is unfavorable. Unfavorable. Okay, so in general, let's see. So the inner diameter of the helix, 4.5 angstroms. This is just for you to appreciate the distance the fact that it's so small that there's really nothing that can fit inside. The outer diameter, this will include the side chains, right? So some things can get entangled in between the side chains. And so for example, one important consequence of that, it fits well into the major groove. We'll talk about minor and major groove of double-stranded DNA. So what that means is that proteins in their alpha helix secondary structure can interact with DNA, right? And we know that DNA, for example, expression of various genes is controlled by, for example, various proteins that tell the DNA express this protein or don't express this protein, right? They call transcription factors. So let me write this down, transcription factor. So many of these transcription factors, when they control the expression of DNA, they contain alpha helices secondary structure that they use to interact with DNA. Transcription factors. Okay, what kind of sequence gives an alpha helix with one hydrophobic phase? What do you think about that?
If it had a non-polar amino acid.
So what's the spatial, how would you space them?
Well, if
Every
they had
4.
they would have to be like on facing the inside of the core.
Well, if we want one hydrophobic face, so face means on the outside, right? So
You would do
want
every 4 amino acids.
every 4 amino acids, that's right. Let's say X, Y, and then you put, let's say, leucine here. Again, X, Y, and you put isoleucine here, something like that, right? So if every four amino acids is hydrophobic amino acid, then one entire face of the alpha helix will be hydrophobic, right? So basically in alpha helix, you look at every 4 amino acids and that will tell you exactly what's going on. Stable, unstable, hydrophobic, hydrophilic, right? Because that's gonna be only one face of the alpha helix. All right, so not all polypeptide sequences adopt alpha helices. So small hydrophobics such as alanine, leucine are strong helix formers. Now proline acts as a helix breaker because the rotation around the N-C alpha, so this should be not A, but alpha. C alpha angle bond is impossible, right? Because it's constrained to a ring. But also there's another reason why proline is a bad amino acid to form an alpha helix. Can you think of another reason? So the rotation is absent, right? So it cannot adopt the right angle, cannot adopt the -47. What else?
It induces kinks.
Yes, so the angle is kind of frozen, and so the alpha helix the angle is frozen, and there will be obviously different geometry to it. There'll be some kind of a kink, yeah. But what about the nitrogen? Just think about what holds the alpha. Why the alpha helix is so stable? Just think about in general. Why the alpha helices? Why is it like #1 secondary structure? What keeps all these? Now, the fact that you have positively charged, negatively charged, hydrophobic, and so on and so forth. These are side chains. These are important factors, but they're not deal breakers or makers. What keeps the alpha helix intact, what makes it so stable is actually the hydrogen bonds, right? Because it's every NH of every amide bond will be hydrogen bonded to the carbonyl of amino acid, 4 amino acids ahead of it, right, in the N-terminus direction. What can proline make that hydrogen bond? So this will be like a part of the structure of a proline. What is missing here that will not allow for the hydrogen bond to form?
The hydrogen on nitrogen.
There is no hydrogen on nitrogen. That's right. No hydrogen. So proline is not the only amino acid that has a ring. There's a consequence of that. As you take the second position on a nitrogen, you replace the hydrogen by an alkyl group. So proline is incapable of making a hydrogen bond to the carbonyl of the N+4 amino acid, right? So that will make the alpha helix very unstable. So write this down. So proline acts as a helix breaker because the rotation around the NC alpha bond is impossible. Plus no H bond. Plus no H bond. What about glycine? Why is the glycine helix breaker?
Because its R group is just in hydrogen.
And what does that do?
Allows
Why is it bad?
other molecules to interact with it.
Well, it makes it very flexible. So if we go back to the structure here with all these R side chains nicely arranged on the periphery, right? So alpha helix allows, one other benefit of alpha helix is that it prevents stereohindrance. It puts all these R groups to the sides on the periphery, right? It spreads them apart. It basically prevents steric hindrance. Now, if you have a glycine, you lose that kind of benefit. So glycine will not benefit from this kind of stuff, right? It doesn't have an R group. And so in glycine is very flexible. It can support any other kind of conformation. So the glycine may say, why do I need alpha helix if I can adopt something else? I'm very flexible. I don't have the R group. I don't need to worry about steric hindrance. And I can take up something else. All right. And so we just talked about attractive or repulsive interactions between side chains. 3 to 4 amino acids apart will affect the formation. We talked about that. Let's see if there's anything else we want to, any other, hold on, there's, yeah, very important. So couples jump, okay, what's this? Propensity of amino acid residues to take up the alpha helical conformation. Okay, so this is what we just talked about. So if the alanine is the best one, right? So then, so this is the energy which will count against us, right? So the energy that you will raise the alpha helix by replacing alanine with arginine, for example, right? So replacing alanine with arginine, you go up in energy by 0.3 kilojoules per mole. So asparagine, cysteine, quite a bit, right? So these are just average numbers. Again, especially with the charged ones, right? It totally depends on what amino acid you have in the N+4 position, right? So I guess this is just general number 0.3, because in general in alpha helix, if you have arginine and then N+4, you will probably see glutamate. And you can see glycine basically doesn't do anything, right? So you by replacing alanine with glycine, you raise the energy by quite a bit. So you lose a lot of this benefit of no steric hindrance. And what else? You can see that, and proline is even larger than 4, right? Larger than 4. No hydrogen bonding, as I said, the kink in the alpha helix, and so on and so forth. All right. So there is one other factor that we need to consider, something that can support alpha helix. If you look at the alpha helix in terms of static charges, so the carboxyl terminus, right? So here we have a COO minus. The amino terminus, here we have NH3 plus, okay? And so these charges, so you can see here on the top of the alpha, on the top of this ribbon of the alpha helix, well, this positive charge will be propagated through the entire alpha helix chain, and the carboxylate will give it a negative charge, which will be propagated throughout the entire alpha helix chain. And so what that means is that you want to, let me write this down. You want to position, since there is a charge separation, you want to position positively charged amino acids around the carboxyl terminus and negatively charged amino acids around the amino terminus. So they will stabilize this charge separation. So, or the helix dipole. Okay, let me write this down. So here's negatively charged amino acids are found. Right? And here is where we have positively charged amino acids are found. All right, I think I've given you quite a lot of things to look for in terms of what will be favorable and unfavorable for the formation of the alpha helix. And you should be able by now look at the primary structure. Let's say I gave you a linear structure of, let's say, 12 amino acids, right? And I ask you, will this adopt the alpha helical structure? And you should look at all these factors, right? N+4 amino acids, what's there? Positively charged, negatively charged, hydrophobic, non-hydrophobic. Also this helix dipole, right? So you can look at the carboxyl terminus, and if you see a lot of positively charged amino acids there, negatively charged amino acids at the amino terminus, if you see all these pointers, then you can say, yes, I believe this will be, this segment of protein will adopt the alpha helical secondary structure. Any questions about alpha helix?
I have a question. So are we saying like, so let's just say that you gave us like this structure that you have right now on the slide, and then you positioned negatively charged amino acids by the carboxyl terminus. Then we would say that it wouldn't form the alpha helix. If like in the linear structure, does that make sense?
Yeah, it will be unfavorable for the alpha helix formation. I'm not saying it will not form it, right? So it's a balance, right? So it's a balance of all things. Now, probably at this point, it's very hard for us to predict which one is more important, right? So, but if I give you all the positive interactions, then you should be able to predict the alpha helix. If I give you all the negative ones, you should say no alpha helix here, right?
Okay, thank you.
Yeah. I will not give you 50-50 situations. Don't worry about that. Even though in real life, that's what scientists are faced with. Always 50-50 situations. It's like, what's more important? Is the helix dipole more important? Or is it the fact that you have too many glycines or too many prolines and so on and so forth? All right, beta sheets. So the planarity of the peptide bond can create structures known as beta pleated sheet-like structures. All right, so side chains protrude from the sheet, alternating in an up and down direction. All right, so here's the one strand, one protein segment, right? So kind of side view. It's not really clear at this point why we're drawing it like this. It will become clear once we draw its partner. Sheets are held together by hydrogen bonding of amide carbonyl groups of the peptide bond from opposite strands. And it's important to keep in mind that there's a parallel and there's anti-parallel sheets. Okay, let's look at those. So this is parallel beta sheets. So these are H-bonded strands run in the same direction. Hydrogen bond between strands are bent. So let's look at that. So this is all parallel. So if we have, let's say, so nitrogen of the amide, right? So the amide group is like this. And the amide group is like this. And the amide group is like this. So that's why they call sheets. So this amide group, through the NH carbonyl hydrogen bonding, will interact with another strand. This will interact with another strand, and so on and so forth. On the other side, now this carbonyl group here, on the other side of the alpha carbon, right? So this will interact with the next peptide bond, right? So they switch the roles. So if this was hydrogen bond donor, this was acceptor. In the next hydrogen bond, this was the acceptor, and this is donor, right? So here's we have again now the peptide bond, and the peptide bond here. So the only thing that changes is that this now is the acceptor, this is donor. Now we'll go back. We'll go back to the original situation. Donor, acceptor, donor, acceptor, and so on and so forth. And so basically the amide bonds, you can see basically this is the interaction between the amide bonds, and it's alternating, donor, acceptor, donor, acceptor, donor, acceptor, and they all run in the same parallel direction. And because of the geometry, they kind of bend. which kind of weakens the interaction a bit, right? So remember, hydrogen bonds, they like to be straight. So this will be parallel beta sheet, top view. Now the opposite, the one is anti-parallel beta sheet, right? So here we have the amide bonds here running in this direction. So this is the amide bond going in this direction, and this is the amide bond going in this direction. And so here we have the hydrogen bond between the, so this will be hydrogen bond donor. This is hydrogen bond acceptor. Now, here the amide bond again. So now, so this is the next amide bond. Now this is hydrogen bond acceptor. This is hydrogen bond donor. But what this allows you to do, there's this quite a bit of space of no hydrogen bond, and then they come in pairs. This pair, this pair, this pair, and the hydrogen bonds. What's favorable here, why anti-parallel beta sheets are so prevalent, is because of the hydrogen bonding that occurs kind of in a straight line, right? So it makes hydrogen bonds stronger. So you can look at both of these slides in more detail, yeah, kind of just to figure out what's going on, like which NH and which carbonyl are bonded. But in general, I told you that what things to look for is the fact that you're alternating donor, acceptor, donor, donor, acceptor, donor, acceptor, and the opposite on the other, on the anti-parallel, right? Here you have acceptor, donor, acceptor, donor, acceptor, donor, and then again, switch, switch, switch again, and so on and so forth. So these are anti-parallel beta sheets. So we will see that actually many proteins adopt this kind of secondary structure, and there are pluses and minuses compared with alpha helices. Again, lots of hydrogen bonding. And you can see the beauty of that is that the R groups are also, what this allows one to do is these R groups are sticking in opposite direction. It's away from us, towards us, away from us, towards us, right? So it also allows to spread the R groups apart and avoid steric hindrance. So these are beta sheets. Any questions about the beta sheets?
Just a quick one, just making sure, just kind of clarifying, the anti-parallel beta sheets are more preferable, right? Because there's stronger hydrogen bonds and the R groups are staggered, I guess. Would that be a better word for it?
You know, I don't really know. Depends on the, obviously, primary sequence. It all depends on the primary sequence. Depends on the what other types of secondary structure are present in the protein, right? So keep in mind that for if you have the anti-parallel beta sheet, it will be something like this, right? This going in this direction, this going in this direction. So this will be anti-parallel. If you have parallel, it will be something like this. Right? So it also depends, besides the primary structure, it depends on which of these is more favorable in the context of the tertiary structure of the protein. When these beta sheets are mixed together with random coils, they're mixed together with alpha helices, right? So which ones, the parallel or anti-parallel, is more favorable on a bigger scale, right? So again, it's a multifactorial sort of answer to your question. There are lots of factors to consider. Okay, and finally, I want to look at, in the remaining few minutes, beta terms. So this will be the third one we're going to look at. So the question is, what happens if you have a, let's say, a beta sheet and it turns around like that? So these are hydrogen bonds. So this is beta sheet, right? Beta sheet. Is this parallel or anti-parallel? Who can tell me? Is this a parallel beta sheet?
Anti-parallel.
Oh, sorry, say it again.
Anti-parallel.
Anti-parallel. So this one is going in this direction, then I turn around, and this one is going in this direction, right? So this will be anti-parallel. So anti-parallel beta sheets oftentimes include a small segment known as beta turns. And you have to have those, right? Because you have to be able to turn around. Okay, so basically we have 180-degree turn, okay? And usually this is accomplished over 4 amino acids, right? So here in this region, we have 4 amino acids. And now glycine and proline come to the prominence. So here, that's where they predominate. All right, so the turn is stabilized by a hydrogen bond from a carbonyl oxygen to the amide proton 3 residues down the sequence. Proline position 2, glycine position 3 are common in beta turns. So let's look at that. So here's we're coming with from our beta turn, from beta sheet. Okay, so this is amino acid 1, alpha carbon. That's the carbonyl of the amide bond. So let's say this is proline. Okay, so proline is in this position. And you can see what happens here is that, so this angle here, what's the dihedral angle between C alpha and N and nitrogen? Is it 180?
180.
Is it 180? No, they're kind of looking in the same direction, aren't they? Remember 180 is when they're trans, when they're looking in opposite directions. Here they're looking in the same direction. So that's what cysteine allows one to do. Okay, it allows basically you to position these in such a way that they actually you can make a very sharp turn. You can make a very sharp turn. So here's the proline. Now here, the third amino acid, here glycine is most prevalent, okay? Glycine, but other amino acids can also be accommodated depending on the group R. And then you go to the NH. And so it's the hydrogen of the 4th amino acid and the carbonyl of the 1st amino acid, which are engaged in the hydrogen bonding. So there is a difference between type 1 and type 2. You can see basically, we're not going to look in great detail in this kind of difference. It's just how this twists around, right? So for example, here from alpha carbon, the next one sticks up. Here from alpha carbon, the next one sticks away from us. Then up, and this is away from us. Right, so basically just different fold, different twist around the amino acids. So no particular rule that we need to know here. But this is the two types of beta turn, and so for proline, it's important to realize that most peptide bonds not involving proline are in a trans configuration, 99.95. For peptide bonds involving proline, about 6% are in cis configuration, right? And most of this 6% involve beta turns. So beta turns are prevalent secondary structures, and most of them will have a proline amino acid. And the reason for that is because it so readily adopts the cis configuration and allows for a sharp turn. Okay, let's look at this picture. So if this is the proline, right? So you can see here, so this is the omega. I was telling you about omega, right? The carbon-nitrogen bond, which has partial double bond character. So it's not so easy for it to rotate. But you can see here, this bond and this bond, the two bonds of the backbone, are at 180 degrees, right? 180. But then what we're gonna do is we're gonna rotate this by 90 degrees. No, we're gonna rotate this by 180 degrees, and we're gonna get into 0 degrees. 0 degrees. Now we have peptide backbone, so this stays the same, in the same place, but this bond of the peptide backbone is on this side now. It was here, right? And now look, it's on this side now. So this has rotated. This has rotated. And so once it's in a cis configuration, now the beta turn is possible, right? It allows for a sharp turn. And there are a family of enzymes known as proline isomerases, right? So it's not so easy for proline to isomerase. It won't do it by itself because of the partial double bond character here. Proline isomerase. So the enzyme will come in and it will flip the proline around its omega, the hydro angle of the peptide angle, from 180 to 0 degrees.
So in order for it to form the turn, it has to be cis?
Yeah. So this is trans. And this is cis. All right, I think we're out of time. So any questions about secondary structures? So we talked about alpha helix, beta sheets, and beta turns. We still have 2 more lectures on chapter 4. I'm gonna expedite a bit on Friday since we still have quite a bit of material to cover. So please use the textbook also to complement the lectures, and obviously sapling. Yeah, so this time you will be pleased. There will be very little math on sapling. Just like on a test, there'll be little math on a test. Any questions? And start working on the practice test. So if you have any questions about the practice test, throw them in a discussion on Canvas and we'll try to address them, right? So I need your feedback. I need your feedback. When I make these tests, you know, I've never administered a test on Canvas before or in an online test, online class. So I will need your feedback. All right, if there are no more questions, I will let you go. And I will see you on Friday. Bye.
Thank you.
Mhm.